Hello everyone, welcome to EPG Pathshala. I am Dr. Shanuja Berry from the Department of Zoology, Kalandi College, University of Delhi. And today I will be talking about the cell membrane, its structure and function. Cell membranes of eukaryotes and prokaryotes serve as a limiting biomembrane with the main function of enclosing the cell contents. Its biochemical composition and structure fulfills various other important roles such as signaling, maintaining non-equilibrium with the extracellular environment, providing a platform for the different functional molecules like enzymes, channels for transport, carriers and receptors, as well as for cell adhesion and recognition. The biomembrane is selectively permeable and very flexible. These two features being due to its structure and composition, and which also contribute to the plethora of unique functions it can carry out. Membranes of almost all organisms have a fluid mosaic organization made up by two main components, the phospholipid lending the basic support and the proteins providing the specific functions attributed to the cell. Structure. The structure of the biomembrane is composed of a lipid bilayer interspersed with transmembrane and peripheral proteins. The composition ranges from 79 to 49% lipids to 21 to 51% proteins. The prokaryotes are surrounded by the single limiting plasma membrane and do not have any intracellular compartmentalizations. The membrane is the site for different cellular functions such as ATP synthesis and membrane transport. In eukaryotes, the plasma membrane is the limiting membrane with various functions associated and attributed to its structure and composition. It differs from that of a prokaryote, as in eukaryotes it forms compartments inside the cell by infolding, and these compartments are known as organelles. The organelles have the same basic structure as the plasma membrane with a unique set of proteins associated with it and dedicated to its specialized function. History. It was proposed by Nageli in 1855 that a membrane surrounds the cell when he observed changes in cell volume when surrounding osmotic pressures were changed. It was shown by Overton that the membrane structure had lipids by demonstrating that non-polar molecules pass through the membrane but not polar molecules. Gorter and Grendel extracted the lipids from erythrocytes and measured the area of the monomolecular layer formed by lipids on the interface of air and water, which turned out to be twice the area of the RBC. They thus established that the biomembrane has two layers, but this study by them did not take into account the presence of proteins in the biomembrane. This fact was presented later by Danieli to justify a lower surface tension of the biomembrane as compared to a pure lipid monolayer at the air and water interface. Electron microscopy further substantiated the presence of a bilayer membrane with approximately 6 to 10 nanometer thickness with proteins interspersed in between. Robertson thus proposed the model of an asymmetric cell membrane due to presence of glycoproteins. Based on these results, the fluid mosaic model of the cell membrane became the accepted structure. The lipid bilayer. The lipid bilayer forms due to the amphiphilic nature of the lipid molecules. The lipid molecules thus have a polar head and a non-polar tail, which can spontaneously form the bilayer structure with the polar head towards the outside and the inside of the cell, and the non-polar hydrophobic tails closely packed and facing each other. The three major classes of lipids present are the ones having the three different kinds of backbones. Glycerol backbone forms the phosphoglycerides, sphingolipids have a sphingosine backbone, and sterols, of which cholesterol are the most abundant. The phosphoglycerides have a glycerol molecule and two of its adjacent carbons each have a long chain fatty acid linked by an ester bond. These fatty acid chains form the hydrophobic end. The third carbon has a phosphate group attached to it, which is further linked to a polar head group, providing the polar moiety. The fatty acid chains can range from 12 to 24 long carbon atoms, and one of the chain is unsaturated. This unsaturation is a cis double bond and can be one or more than one in number. The polar heads attached to the phosphate group can be of different types and are used to classify the phosphoglycerides. A positively charged alcohol is attached to the phosphate group. It forms phosphatidylcholine, which is the most abundant phosphoglyceride in the cell membrane. Phosphatidylethanolamine and phosphatidylserine are some other phosphoglycerides commonly found in the cell membrane and are mostly located in the cytoplasm site. Sphingolipids are formed with a single sphingosine backbone, which is an amino alcohol with a trans double bond between the C4 and C5 atoms. The sphingosine is attached to another fatty acid tail and is now known as a ceramide molecule, and finally forms the sphingolipid. These sphingolipids are of two types, with the phosphate group attached further to a polar head. Sphingomyelin is the most abundant sphingolipid and has phosphocholine attached to the phosphate head. With sugars forming the polar head and no phosphate groups, these are then known as glycolipids and have amphipathic properties. Glycosylcerebrosides have a single glucose molecule attached to the backbone. When the sugar has sialic acid and one or two branch chains attached to the sphingosine, they are called gangliosides. Glycolipids are found on the extracellular surface and form a surrounding which is protective in nature. The third backbone, sterols. The sterols have a basic 4-ring hydrocarbon structure with a polar head provided by the OH group on one end and a short non-polar tail on the other. Animals have cholesterol, fungi have ergosterol, and plants have stigmosterol as the most common sterol in their respective membranes. The cholesterol molecules intercalate between the phospholipids and are found with their hydroxyl group close to the polar head group of the phospholipids. There is one molecule of cholesterol for one molecule of phospholipid. Fluidity of the membrane. It was demonstrated by many researchers and experiments that the lipid molecules show a lateral movement. This was then quantified by the fluorescence recovery after photobleaching or FRAP imaging. In this experiment, cells were labeled with the fluorescent marker which binds uniformly to the lipid molecules. Laser light was then beamed onto a small area which resulted in irreversible bleaching. It was seen then that the bleached area began to show fluorescence again due to lateral movements of the lipid molecules. This migration within the leaflet is very common, occurring at the rate of 10 to the power of 7 times per second and is known as lateral diffusion. Lipid molecules also show rotation around the long axis and have flexible hydrocarbon chains. The migration of lipid molecules, however, from one monolayer to another is rare and is known as flip-flop, carried out by the enzyme flippase. The cell membrane is in a liquid or crystalline state and at a particular temperature changes from liquid to the crystalline or gel state. This change is known as the phase transition. The temperature at which this transition takes place is lowered when, number 1, hydrocarbon chains are shorter, thus reducing their interaction. Number 2, another factor in maintaining the liquid state is the kink in the hydrocarbon chain, which does not allow close packing, and as a result the van der Waals interactions between the fatty acid chains are weaker. And number 3, the third factor contributing to the fluidity of the membrane is the presence of cholesterol, which also prevents hydrocarbon chains from coming together and becoming crystalline. The lipid molecules are not randomly distributed, but are segregated into domains, also known as lipid rafts. These lipid rafts are surrounded by more fluid phospholipids and may contain cholesterol and sphingomyelin. These rafts are microdomains which may contain proteins responsible for transport and receptors for extracellular signaling. Fatty acid synthesis is carried out in the cytoplasm and their transport through the cytoplasm is carried out by fatty acid binding proteins known as FABPs. Cholesterol is synthesized in the cytosol and ER membranes. The lipid molecule is incorporated into pre-existing membranes and cholesterol and phospholipids are transported to different organelles. Membrane proteins. Along with the lipid proteins, the other major components are the membrane proteins, and they can be functional or structural. The amount and kind of proteins are variable depending on the different types of functions carried out by the biomembrane. The proteins too are amphiphilic in nature, having hydrophobic and hydrophilic regions. Based on their functions, the proteins associate with the lipid membrane in many ways and are classified accordingly. Peripheral proteins or membrane associated proteins. These proteins are found on any one side of the membrane itself. They are anchored by covalent interactions to the embedded integral proteins or to the lipids and do not anchor into the membrane itself. They can be released by changes in pH or changes in ionic strength without disrupting the lipid bilayer, unlike the integral proteins. The second kind, transmembrane proteins or integral proteins. These proteins traverse the phospholipid bilayer and have three domains: the cytoplasmic, the exoplasmic and the traversing domains. The cytoplasmic and exoplasmic sides are hydrophilic, but the traversing domain has hydrophobic amino acid residues. Some of the transmembrane proteins may have an additional domain that inserts separately into one layer of the lipid bilayer to increase their hydrophobicity. Some proteins may be located entirely on the cytosolic side and attached by only an amphiphilic domain to the lipid bilayer, such as an alpha helix or by a fatty acid chain. Another kind of anchorage is seen where the protein lies on the exoplasmic side and is attached covalently by an oligosaccharide linker to a lipid anchor, which then inserts into the outer layer of the bilayer. The transmembrane proteins have activities on both sides of the membrane, and their structure is closely dependent on their function. Most of the transmembrane proteins show alpha helical conformation, as the domains that span the membrane only once in some cases, single pass transmembrane protein, or a number of times as in multiple transmembrane proteins. This happens because the polar peptide bonds form hydrogen bonds with each other once in the lipid bilayer. Another conformation that is formed to accommodate the hydrogen bonds is the beta sheet, rolled up as a cylinder, also known as a beta barrel. This is seen in porins, and helices which are aligned together also form interactions, and the protein is folded in this manner. It is these interactions which contribute to the function of a protein. It is inserted into the lipid during the biosynthesis in the ER such that it attains its conformation and orientation based on these interactions and consequently becomes functional. The beta barrels are more rigid and are commonly found in bacterial, mitochondrial and chloroplast membranes. The beta barrel has a lumen which is lined by polar amino acids, and thus it forms a water filled channel. The outside of the barrel is hydrophobic, which interacts with the lipid bilayer and transports small water soluble molecules. This structure is in contrast to the aquaporins, which are formed entirely of alpha helices and transport hydrophilic molecules like water and glycerol. Glycosylation. The transmembrane proteins are glycosylated on the non-cytosolic side. The glycosylation takes place in the ER and the Golgi apparatus. Sugar residues are added as chains of oligosaccharides, which can be either linked to proteins and form glycoprotein or are linked to lipids forming glycolipids. They also form the proteoglycans, which are long polysaccharides attached to transmembrane proteins. The carbohydrate coating is called the glycocalyx and can interact with sugar binding proteins such as lectins, growth factors and antibodies as seen in the blood group antigens. These antigens are related oligosaccharides. What sets eukaryotes apart from prokaryotes, among other things, is the complex division of the eukaryotic cell into various compartments. Each compartment formed by the infolding of the lipid membrane is termed as an organelle. Every organelle has its own specific set of proteins, enzymes and molecules which render it its function. Hence, the membrane not only separates individual cells from each other, but also divides the cytoplasm of each cell into various functionally and chemically distinct organelles. The functions. The many functions performed by the bilayer inside the cell can be approximated in the following ways: structural support, compartmentalization, signaling, non-equilibrium between the cell and its external environment, drastic increase of surface area for protein anchoring and activity, cell adhesion, cell recognition. While the fundamental structure of the bilayer remains more or less the same, this diverse set of function is provided by a diverse set of molecules found in the association with the cell membrane. The plasma membrane exists not only as an external barrier between different cells, but divides, separates or compartmentalizes the cytoplasm of the cell into various membrane bound organelles. Due to the hydrophobic nature, the plasma membrane exists not only as an external barrier between different cells, but divides, separates or compartmentalizes the cytosol of the cell into various membrane bound organelles. Due to the hydrophobic nature of the bilayer, these compartments are impermeable to hydrophilic molecules. Hence, they are mostly isolated from the aqueous cytosolic environment. This provides the unique opportunity of specialization. Depending on the function they perform, each membrane bound organelle is covered in highly specific proteins which control the entry and exit of molecules to and from the organelle. Hence, it is able to control its internal pH, molecule concentrations and other factors for optimal biochemical function. If the cell is examined by volume, the largest volume is occupied by the cytosol. Various organelles are found suspended in this cytosol. The cell nucleus houses the genetic material of the cell in the form of chromosomes. It is the primary seat of nucleic acid synthesis. Continuous with the membrane of the nucleus is the membrane of the rough endoplasmic reticulum. This organelle is almost entirely membranous. It consists of a labyrinth of lipid bilayer which creates a lumen. Proteins are synthesized by the ribosomes bound on the surface of this organelle and are processed and modified within its lumen. The smooth endoplasmic reticulum is devoid of any ribosomes. It is involved in lipid synthesis. Together, the convoluted system of membrane provided by the endoplasmic reticulum consists of nearly half the total membrane content of the cell. This organelle provides vast surface area for membrane bound synthesis of proteins and lipids. Some of the proteins and lipids processed by the ER are sent further into another membranous organelle, the Golgi complex. It consists of various stacked cisternae which dispatch and further modify the cargo en route. The process of ATP production in the cell is also membrane bound. It is coupled with the impermeability of the membrane. ATP synthesis is done by mitochondria and chloroplasts. These organelles again consist of many folded membranes in order to maximize surface area. Degradation on the cellular level takes place at a low pH inside the lysosomes. These organelles contain digestive enzymes which degrade organelles as well as extracellular macromolecules taken into the cell by endocytosis. Endocytosis consists of the engulfment of particles by the cell membrane, which it then internalizes as an endosome. Nearly 50% of the cell volume consists of these membrane bound organelles. Depending on the specialized function of cells, organelle abundance may vary from cell type to cell type. This is especially evident in highly specialized cells. Cells which synthesize large quantities of proteins such as plasma cells or pancreatic exocrine cells contain a disproportionately large amount of rough ER. From an evolutionary point of view, organelles and compartmentalization evolved as the plasma membrane became more specialized. Second function, structural support is provided to the cell by the association of cortical cytoskeletal elements within the lipid bilayer. The cytoskeletal side of the cell membrane is supported by a meshwork of cytoskeletal filaments which play a dual role. They provide a definite structure and foundation to the external cell membrane and act as anchors for membrane proteins. A classical example for this is the presence of spectrin in the cytoplasmic side of the RBC membrane. A long, thin, flexible protein filament, spectrin, is the only cytoskeletal component present in the RBC. It exists as a cytosolic meshwork which is flexible while providing structural integrity. This is extremely important for cells like RBCs. They routinely face contortion and stress while forcing their way through capillaries. Mice and humans with abnormalities in spectrin have spherical, fragile RBC and subsequently are found to be anemic. While spectrin is specific to the RBC, a highly complex and dynamic cortical cytoskeletal support structure is seen in almost all cells. Most cells are supported by actin filaments on the cytoplasmic side of the cell membrane. This cortical network of actin in conjunction with the Rho and some other proteins provides structural integrity to the cell, while being essential for many dynamic processes such as movement, endocytosis, formation of philopodia, lamellopodia, etc. A type of cytoskeletal protein known as lamins are important in providing structural integrity to the nuclear envelope. These lamins belong to a class of cytoskeletal proteins known as intermediate filaments. Another intermediate filament known as keratin is involved in the formation of cell-to-cell adhesions known as desmosomes. The cortical cytoskeletal elements also work towards corralling or dividing the cell membrane into domains. These elements hold membrane proteins in place and restrict protein diffusion across the cell surface. They play an important role in cell-to-cell adhesion as well. Proteins and sugars associated with the membrane play an integral role in cell recognition. The various membrane associated proteins on the external surface of the plasma membrane of the cell do not remain naked on the surface of the cell. These are covered by a coat of sugars known as the glycocalyx. The glycocalyx consists of both saccharides bound to membrane proteins covalently or as proteoglycan molecules. The position and enormous variety of these molecules make them prime candidates for various cell-to-cell recognition processes. Plasma membrane bound lectins have been identified which recognize specific oligosaccharides on the surface of other cells, allowing for cell-to-cell interactions such as sperm-egg interaction, clotting and inflammation. Signaling. The plasma membrane allows propagation of signal molecules to and from the cell. The plasma membrane, through its secretory processes, is capable of secreting specific signal molecules which then go on to interact with other cells. More importantly, this implies that there are specific proteins on the surface of the cell which are capable of recognizing specific signals present in their ECM and are capable of propagating these signals to the various compartments of the cells as meaningful instructions. Signaling can be of various types. Number 1, intracrene signals are produced by the target cell that stay within the target cell. Autocrene signals are produced by the target cells, are secreted and affect the target cell itself via receptors. Juxtacrene signals target adjacent touching cells. These signals are transmitted along cell membranes via protein or lipid components, integral to the membrane and are capable of affecting either the emitting cell or the adjacent cells. Paracrene signals target cells in the general region of the emitting cell. Endocrine signals target distant cells. Endocrine cells produce hormones that travel through the blood to reach all parts of the body. It can also imply various primary and secondary messengers, based on which different types of signaling pathways are known. Example, the JAK/STAT pathway, the PIP2 signaling, the G protein coupled signal receptors, the MAPK/ERK pathway, etc. Presence of specific proteins on the surface of the membrane allows for transport of specialized molecules. Most membrane proteins traverse the plasma membrane in the form of an alpha helix or as beta barrels. They may be single pass, traversing only once, or multipass, traversing the membrane many times. Most multipass helices and barrels interact with each other respectively to form large channels and porine proteins, which allow the plasma membrane to transport materials across itself. A classic example of interaction between helices can be seen in the case of again aquaporins. which consist of hydrophilic core to allow the passage of water through them. Most multipass membrane proteins consist of these alpha helices. These helices can move against each other and are capable of providing conformational change in the molecule to create open and shut ion channels or to transduce signals. The beta barrels interact with each other in a more rigid manner to form larger pores and channels. Some create water-filled channels which transport hydrophilic molecules across the membrane, but not all beta barrels form pores. The lumen of many such protein structures is filled with amino acid residues. These proteins function as enzymes or receptors. The next function, the plasma membrane, is capable of maintaining disequilibrium between the cell and its environment. The plasma membrane can tightly regulate the contents of the cell, creating an internal environment vastly different to that from the external environment. This is best exemplified in the neuron, where the disequilibrium maintained by the cell is used to create a potential difference. The neuron, via active ATP dependent pumping of sodium and potassium ions, creates and propagates an action potential which is the basis of the nervous system. Similarly, the process of ATP synthesis itself requires the creation of a gradient such that the inner mitochondrial membrane pumps protons into the intermembrane space to create the required gradient. Such pumps and transporters are embedded in the membrane and are usually activated by a specific ligand or ion such as calcium cations or acetylcholine. Changes in pH or ionic concentrations also trigger the opening and closing of these pumps, as demonstrated in the propagation of an action potential. So, to summarize, all living cells, prokaryotes and eukaryotes, have a plasma membrane that encloses their content and acts as a semi-permeable barrier to the outside environment. The plasma membrane has a fluid mosaic nature and is composed of lipids arranged in a bilayer and proteins interspersed in between. The lipids are amphiphilic with the polar head and a non-polar fatty acid chain. The proteins are also amphiphilic and are arranged in the lipid bilayer in such a manner that it determines their function. The proteins act as receptors, enzymes, transporters and cytoskeletal support. Transmembrane proteins span the bilayer as a single pass alpha helix or multipass helices and beta barrel rolls. Some proteins are only attached on one side of the membrane by a covalent attachment to the lipid bilayer or can be attached non-covalently to other proteins. Most of the proteins and lipids facing the extracellular side have oligosaccharide chains attached to them which are covalently bound, forming the glycocalyx. Proteins are able to diffuse through the membrane, bringing about transport and other functions. A particular activity of the membrane is conferred by the presence of both lipids and proteins, which get confined into domains. The plasma membrane thus allows the cell to perform its chemical functions in a secure and discrete environment. Thank you.